Okay, hello again. The following slides may seem to show that whatever I want to explain looks very difficult, but it's actually just some basic knowledge that you should know. For example, here again, you know for a fact that DNA is made of AGCT and RNA is AGCU. So here you go. You have your in both DNA and RNA, you have cytosine, adenine, and guanine, and then in DNA you have thymine, and RNA you have uracil. You know, these are the basic structures. You need to understand that the C's and T's are single rings, and then the A's and G's are double rings. You know, and then you have pairing. You know, so these are some of the basic. Again, I'm sorry. I apologize to the international students. This is from an old note slides that I have that actually I forgot to actually change them to English. But basically what it's trying to say is that the attachment of the for purines and pyrimidines is actually attached to carbon number 1, and then it depends on, so again the molecules on the bases are also numbered. Here is the nitrogen 1, and here is nitrogen number 9. So for a purine, is called a N9C1 glycosidic bond, whereas for pyrimidine it is N1C1 pyrimidine glycosidic bond. So that's just it. Here so here you see the difference between a nucleoside, sugar and base, and a nucleotide where you have one phosphate. Nucleotide is also nucleoside 5 monophosphate. Nucleoside monophosphate, you add another phosphate, becomes nucleoside diphosphate. You add another one, it becomes nucleoside triphosphate. So these are the specific names for it. This is just a table. It's not very difficult, and I wouldn't say it's not important, but I need you to understand more than memorize this, you know, because there's no point memorizing if you don't understand it. So basically this is the different types of bases. You have if it's a nucleoside, the ribose plus adenine, it becomes adenosine. If it's a deoxyribose plus adenine, you become deoxyadenosine. Yeah, can you understand that? So for guanine plus a ribose, you'll get a guanosine. If it's a deoxyribose, so it becomes deoxyguanosine. You know, things like that. But you take note that for uracil and thymine, you have very specific names. And if it's a nucleotide plus adenine, so this become ribonucleoside monophosphate. So you have adenylate acid or adenosine monophosphate and guanosine monophosphate, and you know, things like that. So if it is there's a comma there, so that means there are two different names. Either it's acid deoxyadenylate, or in English it's deoxyadenylate acid, or is even called deoxyadenosine monophosphate. You know, so again, I wouldn't go on too much about this. All right. Oh, this is the English version. All right. Okay, and then okay, for pairing purposes, this I think if I find a video on Shagaf's rule, Shagaf is the person who, Erwin Shagaf, did experiments where they isolated different bases, different nucleotides containing different bases, and they found that there is always a correlation. The amount of A's is always the same amount of T's. The amount of G's is always the same amount of C's. And so they're not always totally the same. So that means to say that he says there seems to suggest that there is a interaction between a purine and a pyrimidine. The number of purines is always the same as the number of pyrimidines. Yeah. So this is where you get the AGs over CTs. The number of here pyrimidine over purine. Sorry, this is purine over pyrimidine. The number of purine over pyrimidine is 1. It's always. So from there, and then later when they concluded that DNA is in a double stranded, so this goes to explain how for every purine it must be paired with a pyrimidine, and the pairing is not random. and A must always pair with the T. T must pair with and then A, G with the C, and vice versa. Alright, so here you get the pairing T and A, G and C, C and G, T and A. It's always specific. Alright, but it's always a purine and a pyrimidine. You can never get two purines, you can never get two pyrimidines. So that's why this is a very beautiful molecule with very a regular and a very consistent structure. Some important facts. You have this what you call as the minor groove, and then you have the major groove. Even if it's groove, is actually it actually plays an important role for protein binding. You'll find later that this is where protein specificity, how the protein will actually find certain types of DNA, certain sequences. How can a protein be interacting with DNA is dependent on these types of grooves. This is the minor groove and the major groove. And if you look here, one complete turn is roughly about 10 bases. So you can actually count. One between the distance with one is about 0.34 nanometers, and then the distance between of one complete base is 3.4 nanometers. 3.4 nanometers is the same as 34 angstrom, not Armstrong. Alright, please don't make that mistake. This is angstrom. Alright, and so the 34 angstrom is the same as 3.4 nanometers. Some of the terms that you need to, well, by now you should know what they are. You should be able to say what is a 2-deoxyribose, what is a nucleoside, what is a nucleotide, or nucleotide is the same as nucleotide monophosphate. What is alpha, beta, and gamma? Maybe I'll let you find that out, or if you can't, you probably want to ask me in class. I will not explain what is the alpha, beta, and gamma. What is the 3', 5' phosphodiester bond? What is a pyrophosphate? I think you need to make sure that you know this subject, because yes, I will ask them in tests. Alright. Here's a story about Erwin Chargaff. Okay, I already explained that. Purine is always, the amount of purine is always the same amount of pyrimidine. So adenines to thymines, guanine. So that's why you get AT over GC, AG over TC. A plus G over T plus C is equals to 1. Purine over pyrimidine, because they always pair, so it's equals to 1. However, A plus T and G plus C does not necessarily become 1. This is what they call as, it's just a specific number for different species. This is a report, this is a work by Rosalind Franklin and Morris Wilkins. They did what they call as X-ray diffraction, and what they learned from this is that the DNA is a very constant molecule. The many different groups are trying to figure out the structure of DNA, and Watson and Crick were playing with like building blocks. And that's when they found that if it's all purine, if it's A's and T's, and the only way that you can get that is by if the DNA is actually anti-parallel and the specific pairing. So that's how they came up with this proposal that the DNA is in structure. This is the actual paper by is called A Molecular Structure of Nucleic Acids, published by Watson and Crick in 1953, that won them the Nobel Prize. Some facts, I mentioned this, so it's a 2-chain, that means you have double-stranded DNA, a spiral, right-handed helix. You all know that the chains are anti-parallel. Remember, one is 5', 3', so the other one must be 3', 5'. Right, so if it's 5', 3', the other one must be 5', 3'. It's always like that, regardless of whether it's straight, whether whatever facing whichever way. So the DNA molecule must be anti-parallel. The sugar and phosphate forms the backbone with the bases facing inside. Right? It's like a ladder. A ladder, you have the thing, and then you have the rung of the ladders. That's like that. It's just that this ladder is actually twisted. Okay, so bases occupy planes perpendicular to the axis, stacked one on top of the other. This is the interaction. You have the van der Waals, interaction between the stack bases and hydrophobic interaction. Give it a very stable and very strong structure. The strands, between the strands, the two DNA strands, they are held together by hydrogen bonds. We know that G and C has got 3 hydrogen bonds, the A and T has got 2 hydrogen bonds, and then the chemical properties that shows only A bind with T and G. Again, have a look at the videos. It's easier perhaps if you look at the videos with 3D images. Okay, major minor groove, I've already mentioned that. We'll look at the structure and properties of the rRNA, mRNA, and tRNA later when we look at translation. Okay, so that ends one of the first series slides. I'll see you in the next lecture or video.